Okay. Well, good morning slash evening slash afternoon to everyone, depending on where you are joining us from today. I am delighted to welcome everyone today to our webinar from Marama Labs on making characterization of complex nanoparticle therapy simple. I'm Brendan Darby. I'm the CEO and one of the co-founders of Marama Labs, and I'll be your host for today's webinar. So firstly, just thank you to everyone for registering. It is great to see so many attendees from industry and academia, and a few familiar collaboration faces as well that we see joining us here. So I am very excited about today's webinar, which will focus on the latest advancements in mRNA and LNP characterization, with a particular focus on Marama Labs' scatter-free absorbance technology, also known to many of you as CloudSpec, and its benefits for streamlining LNP characterization workflows. We have two incredible speakers presenting today who will be sharing their insights on CloudSpec and LNP formulation characterization. First up, we have Professor Eric LaRue, co-founder and Chief Science Officer of Marama Labs. Eric will be presenting a deep dive into the physics behind the CloudSpec and how this enables quantification of RNA and ligand concentrations in intact LNP formulations. We then have Associate Professor Piotr Kowalski from University College Cork in Ireland, who will be presenting an overview of the results from a collaboration between Marama Labs and Piotr's group. And those results were recently just published in Analytical Chemistry, demonstrating how scatter-free absorbance overcomes some limitations with traditional fluorescence-based assays for characterization of next-generation LNPs. So just a few short words for housekeeping. We will have 5 minutes for Q&A after each presenter presents, and we then should have a few minutes towards the end of the webinar for any outstanding questions. You can post your questions in the chat directly. So to do this, press the chat button at the bottom of your screen in Zoom, and then on the right-hand side of your screen, the chat window will appear, and in the top right, you will be able to choose New Chat and chat with the host, so you can post your questions directly to me. We'll try to squeeze in as many questions as possible to the speakers within the time. Okay, so finally, the webinar will be recorded. It is being recorded, and we will share this after the webinar. We'll share the link. It'll be posted on our website. So if you do need to leave early, you want to rewatch it or share it with your colleagues, the link will be available. Okay, so without further ado, we will hear from our first speaker, Professor Eric LaRue. Eric is the Chief Science Officer of Marama Labs and Professor of Physics at Victoria University. He was my PhD supervisor, I'm very proud to say. He's the author of over 70 papers in nanophotonics and electromagnetic modeling, and he has over 24,000 citations and is a leading contributor to the field of surface-enhanced Raman spectroscopy. Eric, over to you.
Thanks, Brendan, and welcome everyone. So yeah, as Brendan said, I'm the Chief Science Officer at Marama Labs, originally from France, now based in Wellington, New Zealand. I will, in this presentation, give a brief overview of scatter-free-absorption spectroscopy, which is essentially the technology underpinning the CloudSpec instrument. And I will focus also, obviously, on the applications to RNA and antibody quant in LNPs. So I wanted to start with motivating why did we develop this technology and this instrument. And many of you would be familiar with standard UV spectroscopy. Think your Curie or nanodrop in your lab. And this is a standard method whereby you measure transmission in a sample over a known path length. So look at how much light you have out compared to how much light in, typically having a blank and a sample. The light will decay exponentially, and therefore we take the log, and it's standard that according to Beer-Lambert law, that absorbance quantity will be proportional to concentration. So you can use this to determine the concentration of your sample. And typically it can be used using absorbance at 260, for example, for RNA, or DNA, and at 280 for proteins. So you have a nice peak increasing with concentration, and you have a nice linear dependence. The slope, called the extinction coefficient, allow you to go from essentially a measured absorbance to a measured concentration. What's not often clear is that what you measure in extinction is not really the absorbance, but what's called the extinction. So this number here should be called the extinction. And why does it matter? In the context of LNPs or many other type of samples, once you have particles in your sample, so once your sample is not completely clear, those particles will scatter light. And that means that this light is not collected in your transmission measurement. So overall, we're not measuring absorbance, we're measuring extinction, which is the sum of the absorption contribution, which we care about for Beer-Lambert and concentration, and a scattering signal, which typically is fairly broad and particularly strong in the UV. So this is a fairly common situation, which essentially prevents the application of UVVs to turbid solutions. And there are many in many contexts. Think of your preferred hazy pale ale or beer, and milk is the perfect example where you can't see through milk. When you measure the UVVs of milk, you have essentially nothing, no light going through. And if you measure it, for example, in CloudSpec, where you can see the true absorption, then you start seeing the chemical information of your sample. Okay. So going back to LNPs, this is a typical example of why there are problems with UVVs of LNPs. Your blue light here is what you measure in your UVVs. It is a superposition of the true signal from your pure RNA times a background, which you can essentially see this. There should not be any signal here, but we have this scattering background. Okay. Now, what we can see is that background typically goes up like that as you go to the UV. Okay. So one way of trying to use UVVs for this is to try and essentially guess what that background would be. And there's been quite a lot of work on this post-processing or scatter subtraction algorithms on how can you use UVVs to still recover the true absorption. And it's worth looking at this in more detail. So the principle here is that you take a region with no absorption, like here, and then you choose a scattering model. And there are several in the literatures. This is one of them, but there are others. And then you extrapolate your scattering, essentially knowing what it is in this region, you extrapolate it to the lower region. Once you've done this, you can just subtract and try to recover an absorption spectrum from which you can deduce the concentration. And there are a few issues with this approach, but the main one is that this scattering model is not very well defined. There are various versions of it. And you can have two scattering models that look very similar in the region here, but are very different in the region there. So you might have a small error here of a few percent that transform in a big error here in the region of interest. So that's why it can be very tricky to get accurate results with this method. There are a few other issues in the context of LNPs. Many lipids absorb up to 400 nanometers, so that pushes up the region from which you extrapolate quite far off in the visible, making things worse. It is known that the scattering spectrum depends on size, internal structure, and I've got here examples calculated from Smith theory for different size and internal structure, and you see they all have the same scattering in this region, but they all differ in the region of interest. So essentially, we don't have a way of finding which model to use for a given type of particle. We talked about this, the extrapolation magnifies errors, and this is an example here where so in the CloudSpec, we can measure the true scattering spectrum, and you can see that if you choose one model versus the true scattering, you will get quite a big error in your resulting absorption spectrum. Okay. Now, there is one glimmer of hope here is that those two spectra don't look exactly the same. Okay, so you could think of improving those algorithms by comparing your results to essentially a reference spectrum, what you think you should get, like your pure RNA, And then you could iterate and try a different scattering model. So you could play with the scattering model until you get something that looks as close as possible to what you expect. Okay. And in this case, it works quite well. Because we have just a single component, we know that we expect RNA, and therefore we know that this one looks better than this one. Okay. The problem with this is that as soon as you have more than one component, so imagine that your lipids absorb or you have antibodies, then essentially with two components, you can make this agree almost all the time. Okay, so it means that you lose the ability to distinguish which one is the correct one. Also, what we've observed is that sometimes, the LNPs have a slightly different spectrum than the pure RNA for many different reasons. So this approach would fail because you will try to tweak your scattering model to make it look the same as your reference, but in reality, you're not really, your absorption doesn't look like your reference. Okay. And overall, if you use this approach, you can't tell, you will always get a fit that looks good because you will tweak the scattering model to make it look good, but you can't tell if something went wrong or not. So really what the SFA promises is to avoid all those problems by really not, we don't want to make any assumption of the scattering. We just want to measure the absorption spectrum directly. So we have a physical way of measuring the absorption spectrum directly, and we can spot all those problems. Does it change? Does it shift, etc. So how can we do this? The main idea is to use what's called an integrating sphere. It's a cavity that traps light, and you put the sample inside the cavity, and all the scattered light essentially gets trapped in that cavity. So whether your samples scatters or not, it won't matter to what the light you detect. Okay, so this means that you get something that's independent of scattering. Now, relating it to the true absorption is a bit more complicated because the light bounces around your cavity randomly, and you will go through your samples many times, and you have essentially an effective path length. And a lot of the work, under the hood in the CloudSpec is to correct for that effective path length. Okay. The effective path length is actually quite large in the CloudSpec. At low concentration is of the order of 4 centimeters. So we actually able to measure lower concentration of RNA with better accuracy. This is how it's implemented in the CloudSpec. We actually have 2 lamps and 1 spectrometer so that we can do this SFA measurement and at the same time do a normal transmission measurement. Okay. We have a way of essentially inserting the sample in there. And what's important is to have a constant volume here because obviously if you have more samples, the light will go through it more times. So it's very important to keep a constant volume, which we use 1 milliliter. So the CloudSpec measures those 3 things. Standard UV-Vis in this transmission, absorbance using the integrating sphere, and then we can get the scattering by taking the difference. And this is an example using SM102 nanoparticles, LNPs. We have a clean absorption spectrum that look very much like the the pure RNA in buffer. We can measure empty LNPs to show that in the case of SM102, they don't absorb. Here, they have barely no absorption. And at the same time, we can measure scattering spectra. And you can see the scattering of the empty LNPs is quite different to the scattering of the loaded LNP. So usually they have different size and things like that. All right, so now I will discuss briefly how we can do RNA quantification in practice using SFA. So the most common case I've shown it to you is the one where we have non-absorbing lipids like SM102. In that case, we can just measure A260. There's no absorption from the lipids, so all this comes from the RNA inside the particle. And we use standard methods like normal UV-Vis. We know it's proportional to the concentration. We can deduce the concentration. If we use standard extinction coefficient, in this case here, we can deduce a concentration of 16 micrograms per milliliter. If this change depending on the type of RNA, so you can be more precise by measuring some pure RNA of known concentration. In a CloudSpec, this is the range where it works. It's simply we want to have enough signal, but not too much, like in normal UV-Vis. And typically, we have to use dilution to go down to this range. In practice, what this means is that if you start with fairly highly concentrated RNA, then you would use a fairly high dilution factor. And the material, how much material you use, given this limit, is about 2 micrograms. So you will need 2 micrograms or 2 microliter of those high concentrated RNA in order to detect it with this method. And as a bonus, you get scattering spectra, which can be used to compare, you can relate them to the particle concentrations or particle size. We have validated this, and if you're interested, you can refer to this paper. Essentially, we tried mixtures of RNA and empty LNPs to make sure that we could get scattering independence, we could get good linearity, and we also compared the results of actually encapsulated RNA to orthogonal methods. RiboGreen, but RiboGreen had quite a big a large uncertainty, but we also had a combination of HPLC and RiboGreen, which had a lower uncertainty, and we had good agreement for at least 2 types of RNA. So from this study, essentially, we concluded that the precision of the method is very good, and the accuracy is also better than typical RiboGreen accuracy. So you get the convenience of a simple measurement, but you also get very high precision and good accuracy. It also works with absorbing lipids. So MC3 is the a simple example of an absorbing lipids. This is the absorption of the empty LNP. So it has quite a bit of absorption in the region of the RNA. So in this case, we need to do a deconvolution. We have an RNA reference spectrum, which you need to measure once, and same for your reference lipids, you need to measure them once, typically an empty LNP, and then you can reuse them for all your deconvolution for one type of lipids. And then the deconvolution is very well constrained because we have only 2 parameters, 2 spectra, 2 parameters, and basically it gives a very, very good fit with really a high precision. I should mention here that if you try to do this sort of 2-parameter deconvolution with the scattering subtraction correction, things go wrong because you add at least 2 more parameters in your scattering model, which type of model and how strong the scattering is. And this sort of fits with 4 parameters become highly uncertain and unreliable. If you have enough parameters, you can fit anything. That's what we usually say. Yeah. Okay, and I will finish with an application that we've looked at more recently, which is how we can combine this technique to also measure antibody that is attached to the LNPs. Now, the antibody, it's a type of protein, so they can be quantified typically by their A280 signal. We have an example here. You can see that those 2 spectra are sufficiently different that a deconvolution algorithm would work. And so that's basically the underlying idea. You measure the mixture and you do this deconvolution. As before, you need a good reference spectrum, and typically you need to measure it once for one type of antibody and then keep it for all your deconvolution. One complication of the antibody case is that you can see here that the concentrations are very different, right? So to get a comparable signal, you need an antibody of 100 micrograms per milliliter here. So in typical applications, it's more like 1-to-1. So this is how it would look like. You have a very small contribution from antibody, and the mixture here, doesn't look very different to the actual RNA itself. If you have a 3-to-1 mixture, it's even worse. The antibody contribution here is tiny. Okay, so this is where it's very important to have good reference spectra and to do a deconvolution with only 2 parameters. If you got more than 2, this small contribution will never be picked up. So if you add a scattering model here, it's almost impossible to pick up that small contribution. Even for the one-to-one mixture here in this plot, I compare the scattering model by itself to the scattering plus the antibody. You can see that, this scattering plus antibody is within the error of the scattering model itself. So it will be completely buried if you try to do a deconvolution. Another complications, what we observe with CloudSpec is that a lot of the antibodies are actually scattering. They are big enough to have a bit of scattering. This is an example with real mixtures. We change essentially the ratio antibody to RNA. The deconvolution works quite well. We have very good fits with very good R squared in all cases. And this is the comparison so here we know the nominal because we mixed those RNA and antibody, so we know what they should be. We know what we measure, and essentially they agree quite well. Typically, for these difficult cases where the antibody can be very low, we can get probably a precision of 10%. It's better than that when the antibody has a higher concentration. So this is essentially what we think is the range of applicability. You can probably push this limit, RNA over IB ratio, but you lose a bit of precision. And we have some validation with real conjugated LNPs. We did some experiments. So what I'm going to present here is data from Johanna Simon from Merck, Germany. They were one of the first group using CloudSpec for antibody quantification. And basically, these are the spectra. We have conjugated and unconjugated particles. So those particles have absorbing lipids. So it's much better to use unconjugated as your reference. So we use this deconvolution so that we have only two parameters and to get better accuracy. This is our reference antibody. The deconvolution works very well. Again, some very high R squared. These are three repeats of both the conjugated and the unconjugated. And these are the values that we obtain. And they compare very well to the BCA assay for antibody. We have a very good standard deviation here, probably because it's a one-to-one ratio between RNA and antibody, and also because of the quality of the data here. This is a bit larger, but this is more an assumption on what the unconjugated, the RNA concentration in the unconjugated particle was. So overall, it seems to agree very well with the BCA assay. I mean, the BCA assay has a much larger precision than what we find here on the CloudSpec. So I will just leave you with the conclusions. So SFA can be very useful for this type of applications. RNA quants, antibody quants with some limit on this ratio of RNA to antibody, just because of the very low contribution of the antibody in the spectrum. And these are the main advantage of SFA. And that's my email if you have any questions, after the webinar. Thank you for your attention.
Eric, thank you very much. Very exciting to see the results of what is possible on the latest types of LNPs and formulations. I have not seen, if anyone does have any questions that they're looking to ask, please post them in the chat. We have one question here. Does this work with FABs? So I guess different types of antibody, Eric, and that was a question I was going to ask as well, was does this just work for the type of antibody you've shown, or does it work for different types of antibodies and different sizes?
Yeah, I mean, it would work. So far, we've tried a lot of different types of antibodies, and it works because they all have a similar spectrum, right? So the reference spectrum I showed, you see it here is one type of antibody. This one here that I had, that's one type. You can see it peaks at 280. And there's another type here in Johanna's work, and it has a very similar spectrum. They all look like essentially protein spectrum. So any mixture of protein-based compound would work. Yeah.
Very good. And then that brings you to my next question, Eric. Does this work for any type of RNA, and does it work for different types of payloads like DNA as well?
Yes. So in principle, yes. We've not done too much testing with DNA so far, but the DNA, I mean, we've done DNA quantification without antibody, and it works. The spectra are very similar. So, there's no reason why it wouldn't work. You need to just take a reference DNA rather than a reference RNA and adjust your extinction coefficients. But yes, it would work for DNA.
Gotcha. We've got a couple more questions coming in here. One is, what is the absorbance threshold, in brackets, wavelength and optical density, to consider an absorbing lipid?
Yes. Yeah, that's a good question. I guess it's relative to your RNA concentration. So typically, SM102 has very little, So for example, this one has a bit more absorption. You can see in the empty particles here. So it's, I don't think there's an absolute answer to this. Typically, we will do the analysis. We always measure the empty LNPs, and we do the analysis with the empty LNPs and without, and look at the difference. And in many cases, the lipids don't contribute significantly. Yeah, but there are some like MC3 is one that is definitely strongly absorbing at the concentrations we use in typical LNP formulations. Yeah.
And I think, am I right that there's some demonstration of that in the Nanoletters paper where we have absorbing lipids, Eric? Yes.
We have two types of absorbing lipids that are quite strongly absorbing in the Nanoletters papers.
Yeah.
Yep. And I think Piotr might have also other examples of absorbing lipids. Yeah.
Great. So we posted that link there to the paper, which everyone should hopefully be able to ask. I'll take one more question for now, and then we might take some of these questions towards the end as well. The question here is, Eric, can I assume CloudSpec test free RNA in the formulation for calculation of encapsulation efficiency?
Sorry, can you repeat that?
CloudSpec test measure free RNA in formulations for calculation of encapsulation efficiency?
Yeah, so by itself, no. But we're developing a method, based again on using RiboGreen so that it can be done within the CloudSpec. So at the moment, no, but hopefully by early next year, we'll have something that can be used using RiboGreen, but without needing the lysis and the Triton. Yeah.
All right, great. There's more questions coming in, but we might push on to the next speaker. So Eric, if you're happy to stop sharing, and Piotr, you can start, I'll shift over. So I'm very pleased now to introduce and invite Associate Professor Piotr Kowalski from the School of Pharmacy at University College Cork here in Ireland. Piotr's research focuses on advanced therapy, medicinal products, and nucleic acid delivery, spanning LNPs, polymeric systems, RNA-based therapeutics for cancer, vascular disease, inflammatory indications. He's co-authored several highly cited reviews and primary papers on RNA delivery technologies and smart biomaterials, and he's a corresponding co-author on the recent analytical chemistry study on scatter-free absorbance for nanoparticle formulation characterization published with Marama Labs. So Piotr, without further ado, over to you.
Thanks so much, Brendan, for the invitation. It's really a pleasure to share some of our recent work with you guys. So my lab is focused on engineering clinically relevant RNA nanotechnologies that we aim for high medical need diseases such as sepsis or IBD. So today I will tell you a little bit about our work, and how we try to overcome those challenges by developing novel, more complex delivery systems. But maybe more importantly, I will discuss, how we think that improving the characterization method and like RNA quantification with methods such as scatter-free UV absorption spectroscopy is actually very important to push the RNA therapeutic field forward. So in contrast to conventional drugs, RNA therapy allow precise control over gene and protein expression. So for example, short interfering RNA can transiently silence the desired gene by binding to messenger RNA transcript. And the messenger RNA, for example, allows to produce any therapeutic protein in the host cells. This could be intracellular protein, secreted protein. And in some examples, we could use a protein like this, the one depicted here, which is Cas9, that combined with yet another type of RNA, this sgRNA, single guide RNA, can actually lead to permanent changes into genetic information. So even using two types of RNA, like siRNA and mRNA, we have a very powerful toolbox already to modulate protein or cell behavior and protein expression and gene expression in the cells. And this is a very exciting time for RNA therapies, as you may know, since many of these drugs are entering the market. We have now 7 siRNA drugs approved since 2018, so it's about 1 siRNA drug per year. And we've also seen 3 mRNA-based drugs, primarily vaccines at the moment, also making a huge impact on sorting things like pandemics, most recently COVID-19. Our group in particular also has an ambition to expand that toolbox. We work with different types of RNAs that provides an additional features like circular RNAs. I won't focus too much on this one today, but regardless of the type of RNA that we want to use for therapy, the therapeutic utility is really highly limited by the effective means of delivery of these RNAs to the target tissues. And at the moment, we are really effective in delivering RNA therapeutics to tissues like liver and some of the immune cells. So there's a really high need to develop delivery systems that enable this extra-hepatic delivery to fully capitalize on the potential of these RNA therapeutic drugs. And I think it's important to talk about the evolution of ionizable cationic lipid to really showcase how important, developing these materials and LNP formulation is to push these RNAs towards the therapeutic realm. So here we see an evolution of the ionizable lipid called DLIM MC3-DMA. This is main component of the first siRNA drug that has been put on the market in the form of lipid nanoparticle, which is called Onpattro, and have been actually dosed to patients that have developed transthyretin amyloidosis for nearly a decade already. So it's very I say kind of a crown example of success of the RNA therapeutic in the form of LNP. But what's important here that there has been several structural changes made to those lipids, like kind of iterative changes, introduction, for example, of unsaturation into the alkyl tails, changes in the ester structure, and fine-tuning of the pKa of the lipid head group that actually led to over 4 orders of magnitude improvement of the therapeutic potency of that lipid for gene silencing in the liver. So this is really a showcase case study, which actually evolution of the lipids is really critical to push the LNPs for delivery into the therapeutic kind of ranges. And this lipid has been dosed to patients actually every 3 weeks for over 5 years in some cases. So it's been very effective therapeutic strategy, even for like a chronic administration. So here you see a couple of other examples of lipids that has been actually a cornerstone for developing RNA-based therapeutic drugs, in this case, ALC-0315 and SM-102 that you heard about. These went both into Moderna and Pfizer vaccines. And what you see here, you can see the further changes into the lipid kind of structure, in particular, this branching of lipophilic domain, introduction of this protonable amine and hydrogen bonding groups like OHs, really fine-tune this lipid base structures to work more effectively for larger RNA payload, such as messenger RNA in this case. So LNPs are currently leading technology for therapeutic use of messenger RNA and siRNA. But actually, if you look closely enough, for some of the ongoing clinical trials with LNP, we still see with this new generation of lipids that the doses, the primary actually indication of these therapeutic trials are still in the liver, and that the doses that we are using, in particular for messenger RNA, are still relatively about twice as high as for on Patra formulation. So this tells us that lipid nanoparticles are really pushing the boundaries for delivering into the liver, but especially for administration of to extra-hepatic tissues and especially diseases that might require chronic long-term administrations. We're still probably not yet there, and we have to think about utilizing different materials as well as maybe more complex formulation than only the lipid nanoparticles depicted here. So our group actually is interested in developing and utilizing polymers and polymeric nanoparticles to exactly just to improve extrahepatic delivery. And polymers historically has shown promise to deliver RNA to and other nucleic acid cargos beyond liver tissue. They show significantly less liver tropism compared to LNPs. However, they haven't entertained yet such a clinical success as lipid nanoparticles. And this is for good reasons, obviously. So we take, while we're developing new types of polymers for RNA delivery, we're really looking at addressing these critical issues that polymers have kind of suffered over the years, namely efficacy of the delivery for larger type of payloads, biocompatibility. So instead of utilizing cationic materials, we look into more ionizable materials that can undergo degradation and degrade into products or metabolites that would be well tolerated by the body. And most importantly, we also looking at chemistries that allow us to control polymer molecular weight, which is a defining parameter of the material. And this is important also for batch-to-batch reproducibility and scalability. So in my lab, also we work with different types of polymers, and I'll just give you an example of one here, which is this amino polyester that you can see here. This polymer is actually synthesized by a ring opening polymerization of lactones, which are natural product or present in natural product. They use as a food additive, so should be relatively safe as metabolites in monomer. And this is initiated by the tertiary amino alcohol, this ring opening polymerization that gives us this kind of ionizable characteristic due to the presence of a tertiary amine group. So even with the simple design, this two-component system, we can actually control polymer kind of structure. We can control its ability to self-assemble and hydrophobicity, and we still have ability to modulate further post-polymerization the particle properties by post-polymerization functionalization. And we synthesized over hundreds of these polymers in the lab. Majority of these polymers show very good and narrow molecular weight distribution, which is important. And we also proved that we have good control over the molecular weight of these polymers simply by changing stoichiometry. And this is showcased here by this kind of linear relation between the number of repeat units in the polymer that we desire to have and the actual polymer molecular weight that correlate very closely. And since lipid nanoparticles is the most clinically advanced system, we really wanted these polymers also to work in the context of lipid excipients. So what we tend to on Stride to do is actually we want to kind of take out the ionizable lipid and replace it or substitute it with the polymer into the kind of LNP composition. So here you see one of the simple screens that we've done with using firefly luciferase mRNA in cancer cells, and you see that out of the library of about 40 polymers, we found a number of them that are able to deliver RNA intracellularly. Most of these top performing polymers that I highlighted here form very nice nanoparticles, about 100 nanometers in size. But what's actually were quite intriguing, we found that when we looked at cryo-TEM, so morphology of these nanoparticles, we found very interesting structures, and we really found the ability of the polymer and the nature of the polymer shaping how the structure and the morphology of these nanoparticles looks like, which is very distinct from what you might be used to seeing with lipid nanoparticles that despite different lipids may look the same. So here you see very distinct kind of patterns or structures that polymers kind of enforces. So we don't know exactly yet how that translates into potentially efficiencies or tropics, but there is definitely a growing appreciation of how the morphology and structure of nanoparticles can affect, variety of sorting of nanoparticles to different organs or interacting with different cells. So if you're interested in more details in that, we've published recently a paper in Applied Biomaterials here that delves deeper on design of the polymers into lipid nanoparticles. But for the time constraints, I won't be able to focus on that today, and want to move on actually to the main topic. Before I go too fast, maybe just to show you kind of an in vivo performance of some of these polymers, where actually we found performing at a very high level. So about 10-fold higher expression than in vivo JetPi, which is kind of gold standard. In this particular case of the polymers, we were achieving good transfection also in the lung. So now moving on to the main topic of today, which actually the characterization of lipid nanoparticles. So as I said, in my group, we work with kind of unconventional materials with polymers, polymer hybrid nanoparticles. We also utilize materials like dendrimers, and there's a good tradition of people working also with materials like cyclodextrins or amphiphilic cyclodextrin in the School of Pharmacy. So actually, when we actually often see kind of unusual things when we apply the RiboGreen for these nanoparticles, and we are very interested when we met on one of the conferences, the Marama Labs, so namely Brendan and Eric, and we talked with them about the new methods that they are developing for characterization and nanoparticles, because we think that there is really a need to improve even the RNA characterization, especially for complex nanoparticles. So typical way to characterize RNA concentration would be using fluorescent-based methods like RiboGreen that we've heard about. These methods are pretty much gold standard for the lab. And if we want to go to more sophisticated methods, obviously there is methods based on HPLC, for example. However, those required more kind of technical expertise, more sophisticated equipment. So not every lab can actually do it very easily, and they have their own challenges. But typically, RiboGreen is quite easy to do. However, it requires nanoparticle disruption. It's a destructive method, so you cannot reuse the particles or the sample later on. And as we found out many times, and also in this paper, that it has limited precision. In contrast, the scatter-free absorption spectroscopy is non-destructive. Like Eric said, it's shown to be precise, and it's actually quite fast and user-friendly, as you don't need to make standard curve. And if you're familiar with the spectroscopic measurement, it's really easy to utilize. So as I mentioned, the typical fluorescent-based method that we use to reference the reference in this paper, the CloudSpec results, where RiboGreen that requires detergent, usually disruption of the lipid or any type of nanoparticle. Since it's a membrane impermeable dye, RiboGreen cannot access your RNA until the particles is actually fully disrupted. However, we thought about also alternative methods that may actually improve quantification. So we actually use this Cyto9 fluorescent dye, which is membrane permeable. So this can actually go inside, technically should be able to go inside and penetrate through the nanoparticle membrane, bind to the RNA, and provide the signal from your kind of encapsulated RNA. So we really tried hard to battle test the CloudSpec, again, different reference fluorescent-based method, but also throughout different challenging formulations here. But what we found for those fluorescent methods that for RiboGreen, especially formulations that are hard to disrupt, so these polymeric hybrid nanoparticles or cyclodextrin in particular that don't have any lipid excipients, just form complexes with RNA, are actually quite difficult to measure accurately. So RiboGreen can actually underestimate the RNA concentration, total RNA concentration, if those nanoparticles and in the range of 50 to 90%, which is quite substantially. And what you see that LNP that Eric and Brandon has used before, they're actually probably the easiest case study, and RiboGreen is kind of working quite well for them. But looking at Cyto9 that we were hoping to kind of improve things, it does in some cases show improvements. So it can, as I said, penetrate to the nanoparticle membranes. It provides a bit more accurate measurement, still far off from what the expected range of the concentration would be. However, what we also found that these, particularly the Cyto9, is quite unreliable because it's very susceptible to different measurement condition. So student who was performing this work, Ida, was actually seeing a lot of sensitivity to buffer composition, whether it's a PE, for example, or PBS made a substantial difference in the measurement. We also seen dependency on the RNA type and temperature. So there is an interesting there is a potential in utilizing alternative dyes to RiboGreen, but we see that they may have their own challenges, and that's why we wanted to really look at these scatter-free spectroscopy method that offers hopefully an improvement in RNA quantification without destroying your sample. And I won't go into the theory again. I think we had a beautiful introduction from Eric to the technicalities of how CloudSpec works and how this method can be utilized. So I'll just basically glance over the slide, but for someone who is not very technical in the physics of RNA, this method really can deconvolute the true RNA signal from the RNA containing LNP by measuring also the scattering that the particles provide. So when we looked at the spectra for the SFAS method of the nanoparticles, we obviously have empty LNP and empty formulations, and you see majority of them, the formulation did not, including LNP, did not scatter very much the light. We have in particular this one here that is this dendrimer particles, which we included kind of on purpose slightly, because we see that these dendrimers has also aromatic group. So we expected a lot of scattering. So maybe alluding to the question of a highly absorbing lipid, that's probably the worst case scenario that you may have, unless you have many more of those aromatic rings here. But CloudSpec was actually pretty efficient in kind of deconvoluting the spectra for these nanoparticles, as you can see here. And for the other particles that don't absorb too much, we didn't see any problem either with measuring the concentrations. One particular plot I want to also put maybe highlight for you is the scattering of nanoparticles. So as you can see here, different nanoparticles formulation scattered light differently. And again, here LNP had very low amount of scattering compared to some of the formulations that we measure, including these polymers or cyclodextrin nanoparticles. So light scattering, obviously, what we see here is the light scattering, the contributor to light scattering can be either the composition of nanoparticles, but also the concentration of the nanoparticles. And we did some kind of evaluation here, the concentration, but also size, of the nanoparticles. So the larger particles technically could scatter more light than the smaller. So the nanoparticles ranges which we looked at in this particular study range for about 80 nanometers to about 300 nanometers. So with our results, we show that this method can actually be applied to the particles even of a larger size than your typical LNPs and still provide an accurate measurement, and that the particle concentration is also play an important factor here, not only the size. So coming to the main kind of results here with this SFAS, as you can see, utilizing CloudSpec and scatter-free absorption spectroscopy significantly improved accuracy of the measurement for these particles. So now you can see most of the formulations are within really nicely within the expected range of concentration. To make sure that we are measuring the encapsulated RNA, we also perform gel electrophoresis here. So at least we don't have an indication of the free RNA. So these formulations that we're looking at here are encapsulating at a high level. So we mainly measured the encapsulated RNA here. As you know, the measuring the encapsulation with CloudSpec is not possible at the moment. When we contrast this again with the results from the RiboGreen, we clearly see that RiboGreen concentrations are significantly lower than what we measured with the CloudSpecs, particularly for hard to disrupt formulation. We tried actually to improve that, so there can be modification made to the RiboGreen, and IDA tried to use particular heat to improve the disruption of the nanoparticles, and in high heat in some cases, in particular for polymers, resulted in a little bit better disruption. Still not exactly within the concentration range, but for particles like cyclodextrin that don't have lipids, we did not see actually any improvement even when we applied high heat in the RiboGreen method. So overall, what we do see that this scatter-free absorption spectroscopy really provides superior accuracy and reproducibility across all types of nanoparticles. It's a provides robust quantification either to hard to disrupt particles or even to particles that may have a high absorption containing aromatic rings. The convolution that Eric talked about is very effective. It can separate the RNA spectra. In this paper, we used both siRNA and mRNA payloads to get the true kind of RNA concentration, can separate this from nanoparticles and solvent. And I think throughout the study, we also found that this method was kind of very user-friendly. So you really don't have to be very, you don't need a lot of expertise to use CloudSpec. If you're familiar with the spectrophotometry, using a spectrophotometer, this method is not that much different from it. So yeah, it helps with sustainability. You can reuse the sample. So there's a lot of things going for that method, and we really think that this can significantly help to improve, development of the RNA therapeutics and RNA formulation, in particular ones that utilize materials distinct from classical lipids and have complex formulations. So here's my group, and I especially would like to thank our collaborators, so Eric and Brendan and Ida, who's kind of a key player that led the collaboration with the Marama Labs and the perform bulk of the work that I talked about today. So yeah, I'm happy to take any questions, and thank you for the invitation again.
Thank you very much, Piotr, for the very insightful talk and the presentation of the data from the recent paper, as well as an overview of the forefront of the field from your perspective. I guess question from me is where do you, outside of the work here, where do you see, I guess, the most important focus for your group on formulation development is, and where is the future there from your perspective on these new types of particles that you're developing?
Yeah, so I think we need to we see definitely the need to go in order to go for extra-hepatic tissues, we see the need to develop formulations that kind of break the maybe mold of standard LNP formulations, either by putting different types of excipients in them, utilizing a variety of different materials, polymers, blending lipids with polymers. I think this is the LNP technology is great, and I think it will take us far, but it won't solve all our delivery problems. That's, I think, how we think about it. And there's definitely room for a variety of different biomaterials and complex formulations where which kind of will complement RNA technology in the therapeutic space.
Yeah, fantastic. I will say this has been, as Piotr mentioned, a very fruitful collaboration between Marama Labs and Piotr's group. We met at CRS in Bologna a few years back, and then we provided an instrument to Piotr's group, which is there and this continues to be used. So I think it's a good example here of how we are looking to continue to collaborate with academic groups like Piotr's to provide tools that can support advancing the field from an academic perspective and of course an industrial perspective. So if anyone is here from academic groups, please do reach out to us, and I've shared some contact information for Piotr as well as the links to the papers that have been published. So I would recommend you, if you have any other questions in detail on the paper, to reach out to Piotr. So we have a couple more questions that came in beforehand that I'll just try and get through, if that's okay. There was one, just let me find this one here. Yes, so I think this one perhaps is back to Eric. In cases such as prime editing, where two types of RNA are incorporated into the LNP, can the CloudSpec differentiate the two, or does this present too many variables to fit? So Eric, would you be able to?
Yeah, it's not too many variables, I think, but the spectra are probably too similar in many cases. So if the spectra are identical, it won't be able to differentiate the two. It will still work and give the total RNA concentration, but it's unlikely to be able to tell you how much of each type of RNA you've got unless they are really different. The spectra are very different. Yep.
Which is the basis of how the antibody quantification works.
Yes, yes. I mean, some RNA have slight shifts. So if the shift is sufficiently big, like a few nanometers, we might be able to say something. But in most cases, the RNA spectra tend to be fairly similar within a few nanometers. Yeah.
Thank you, Eric. I have another question here. Does Marama have any installed customers who are happy to speak or share experience with their usage of the CloudSpec to date? I can take that one on. Yes, we do indeed. So if you'd like to, you can get in touch with us through the website or to myself, and we can have a call and we can talk you through and share some of that experience from some of our existing customers. You will note that we've referenced here Johanna Simon's group at Merck KGaA in Darmstadt. We've had a webinar that we shared as well back in May with 4Base Bio in the UK as well, who are happy to share their experience. So we're happy to connect you with those offline in the coming days. So please do reach out. Okay, I think that is us. We are just coming up on time. I'd like to we've maybe one more question here. Yes, this is for Piotr. Let's just come in. Given the hybrid approach, what is a possible reason for the tropism more towards the lung and spleen? Is it due to increased zeta potential? That's for you, Piotr.
Yeah, thanks for the question. So no, we don't see the increased zeta potential of the particles. And we do not exactly have the good answer for that. We haven't dived deeper into mechanisms, but it's not the classical sort type of sort mechanism where you kind of see when you substitute a highly charged lipid either negatively or positively, you can shift the tropism. I think we believe that part of the equation here is actually how this polymer changes the nanoparticle structure and surface and modify the surface properties, which might eventually also affect the protein corona. But we haven't performed the mechanistic study to actually show that. There is a little bit interesting hint towards that maybe when you look at the latest paper that we published, where actually a small changes in the side chain of the polymer had actually impact on a variety of different properties and can actually fine-tune, maybe not completely shift, but fine-tune the tropism. So where we think that with so minute changes, you can still fine-tune the tropism, shows that there are probably more of a change to the surface kind of property of the polymer that then governs some kind of interaction with subsequent spotting coronas likely in the serum.
Thank you, Piotr. Okay, I think we will leave it at that. I'm going to stop the recording. Thank you, everybody.